डियर स्टूडेंट्स, लास्ट लेक्चर में हम लोगों ने बायोलॉजिकल डेटाबेसेस के बारे में डिस्कशन किया था एंड वी वर about to discuss about the file formats. So today we are starting with the file formats. So we can see there is a various type of file formats, and first we have to understand what is file format and why we require file format. So each tool and database have their specific file format for better understanding of that particular data. Means when you will come to bioinformatics and you are going to use particular tools, suppose you are going to use some docking tool, so you need a specific type of files to perform, because only those particular files can be readable by that particular tool. Suppose you are going to use QSAR, then you need a specific kind of a file, or you are going to read some database. So in that database you need that database itself has some specific particular file formats. So in each and every tool and database have their specific file format for the better understanding. Why? Suppose if this data is in not in a proper manner, even you cannot understand, or while submitting that data, suppose when a researcher is depositing a data into the database, or that data is not ordered, so that person will not able to understand where that data should be keep posting, or where should they keep that particular data. So it is a very typical task. So to overcome that particular problem, each database or tool follow a particular file format. File format provides the classical representation of data. Means suppose if I'll ask you to just show me a DNA sequence. So a DNA sequence means it is made up of ATGC. There should not be any kind of amino acid, or there should not be a uracil in, like, suppose, which is in RNA. So it is a classical representation that means to that particular kind of data which we are looking for. File formats provides a link between computer and human for the better understanding of data. So a computer should read that file. Suppose when we'll talk about some tools, or some specific format which will be used about some particular tool. So I'll just take an example. I'll discuss in the next slide about some file formats. So suppose a tool requires a specific file format and you will not give that file in that particular format, so that tool cannot recognize that file. Okay. Or suppose a tool will return to a file and you are not able to understand that file, then there is a no meaning of the research, or no meaning of the result of that particular work. Okay, even you cannot understand, or even computer is not understanding. So if both human and computer should understand what is exactly the input and output, so that is the requirement of file format. And we will cover all the 7 types which are in the syllabus, that is FASTA format, GenBank format, EMBL, NBR, FPIR, GCG, PDB, and MSA. These all 7 kinds of file formats I will discuss. I will take one by one, and I'll move further. So first one is a FASTA format. What is FASTA file format? It's the simplest kind of file format. Okay, which so you just look into this particular file. We will come again here later. Just look into this file. So this file is mentioned starting with PDB, and I'm saying that this is not a FASTA format. This file is not in FASTA format. This file is not in FASTA format. So what is the difference you are seeing in this file and this particular file? What is the difference you are seeing? So only difference is this file is starting with a greater than symbol. So this file is starting with a greater than symbol. See here, this is also a FASTA file. So whenever a file starts with a file starts with greater than symbol, that is known as FASTA file format. Anything you can represent, either it is an amino acid, it's a protein file, it's a RNA file, it's a DNA file, whatever it is. Suppose it is AUCRAPST. So this is not FASTA format. As soon as you will add, sorry, as soon as you will add the greater than symbol, this file turns into the FASTA file format. So this is the simplest file format. And one more things I would like to tell you about, it's these things. This is the general description. This is GI, gene identifier. This thing is accession number, sorry, session number, and this is version number, and this is the description of sequence. Okay, but these things are not necessary. You will write these things or you will not. Only thing is this greater than symbol. If this greater than symbol start from here and there is a, suppose I'll rub this, there is no greater than symbol, then there is a no meaning of this file. Tool will not going to read that file. Tool will read the C file from here itself. Okay, so anything, any data which contains a greater than symbol and then the sequence, it is a FASTA format. And then you can see there is a sequence. This area is, this is a sequence and it is in a align manner. If suppose it is terminating here, it is terminated. There is a no terminator. No terminator. You cannot see how it is terminated. It is just a sequence. It starts with greater symbol. That is the unique character of FASTA file format. That is the simplest kind of file format, and it is start with greater than symbol. Hope you understand. Okay. Never get confused with this kind of symbol. No, it is a lesser than. It is this symbol, greater than symbol. You just look into the symbol. If this symbol is there, you can just see and say, yeah, it is a greater than symbol and it is a FASTA format. If nothing is written here, no need of this. If it is written or not, there is a no need of that. Only they are greater than symbol. Okay, so that is FASTA file format. Next is, second one is GenBank file format. Second one is a GenBank file format. So GenBank, as I told you, it is a part of NCBI. It is a part of NCBI. Okay, and it contains three specific feature of their file. Like first one is a header file, header section. Second one is a feature section, and third one is a sequence. So from the very first beginning, from the beginning to this comment section, this is known as header. From feature to this area, before the origin, not the origin, till here, this is known as feature, and here it is from origin, it is sequence. Okay, so this is a sequence. Yes. So this is sequence. So let's begin with this. So first thing is a header. What does header contains? Header contain locus. Here, see this. This is locus. This is locus. So this is header, and header contain locus. You have to remember the thing. It is so many times it is asked how much entries a locus contain. So there is a 6 entries. The very first one is a accession number. I'll explain what is accession number. Second one is a length of sequence. Third one is a type of sequence. Fourth one is a shape of sequence. Fifth one is a organism, and last one is a date of submission. At which what date it is submitted into the database. So let's see what is accession number. Accession number is a unique number. It's just like a roll number. It's just like a roll number. So roll number is a unique number. In a same institute, there is a maybe 10 students or 20 students whose names are similar. Their parents' name may also be similar, but their roll number can't be same. So roll number is a very much unique. It's a unique ID, same like a roll number. So accession number is a unique. In a NCBI database, You might be see if you'll go to the database, there is a million of sequence, but none of the sequence will be having same extraction number. There must be a change. Then there is a length of sequence. What is the length? How many base pairs are there? Okay, so the number of base pairs. Type of sequence, either it is DNA, it is RNA, or it is protein, whatever it will be. Shape of the sequence, either it is a linear or circular. Organism, either it is a human, means primate, or it is a plant, whatever it will be. It is a viral, whatever it will be. It is a letter, three letter code will be mentioned here. Three letter code. And date of submission, at what date it has been submitted. Just see here, it is mentioned here, it is exported. It is this data, this is just a sample file. I'll show you some more files. It is just a exported one, so it is not having extraction number. Okay, it contains 2686 number of base pairs. It is double stranded DNA, circular, synthetic, because it is designed, it is not taken from somewhere else, it is synthetic. And on 24th November 2013 it was submitted to the GenBank file, GenBank database. Okay, so it is very much important. अगर आपसे कोई भी पूछता है कि what is locus, so you can define locus in this 6 1 1 2 3 4 5 6. Okay, that is very much important. This is the main feature of header. From header itself you can get the complete information about the, means from locus itself you will get the complete information about the sequence, what type of bit data, or what you are going to have. Then the same thing it is mentioned here. Next thing is a definition. So it is the name, standard E. coli vector with a multiple cloning site for DNA cloning. The MCCS is reversed in PUC18. PUC18 is a vector. It is it is a synthetic one. It is made using this vector. Accession, there is a no accession number. There is a no version number. If you don't have accession, there is a no version number. So what is version number is, suppose I told you before also, like when I was explaining PDB in the biological databases. So same version meeting is here. Suppose I have submitted this sequence on 24th November 2013 and there is a no accession because it was exported one. Suppose this sequence was submitted which was given accession number, suppose XYZ 1 2 3 4. This was some accession number was given to this particular sequence. Suppose you have make some changes or you make some updation and you are again submitting that data, so you will not be given a newer accession number, you will given a version number. So now your version number is a XYZ 1234.1. again you will make some updates, then it will be like .2, then it will be .3. So as soon as you will make changes, it will keep on updating the version number. Then there is a keyword. So what is the keyword? Whenever we write abstracts, we mention some keywords there. So keyword is feature, what you are going to identify from that. So it's a PUC19. Source, synthetic DNA construct. Organism, there is a synthetic DNA construct. Reference 1 to 2686. It's a complete sequence they have mentioned here. Authors, these two or three authors worked on this. This title means the particular paper in which they have published that sequence. So see, this is the journal Gene 1985 331039. It was published, okay, in this under the title of this. PubMed ID, if you want to look into this particular what to say, research paper, you just go to PubChem, this particular PubChem database, type this ID, you will get the detail about this particular sequence. Then author New England Biolabs, title direct submission, journal, same information is repeating, and comment C also GenBank accession number. See, it is related with this accession number, it is mentioned here. Okay, so by the header portion you can get the detailed by the header folder. So by the header folder you can get the detailed information about the sequence. What is this particular sequence is, you can get the information from header folder. Okay, otherwise it is just a ATGC, what you are going to understand. So the detailed information is available. Next thing comes to the feature. And feature is very important, very important. Okay, why it is important? because feature is contained about the CDS. See here only one CDS is there. Otherwise you will see a sequences which contains more number of CDS. Okay. So what is CDS? CDS is a coding segment. Coding segment. This DNA is a synthetic one, so the coding the size of CDS is complete 122688. Otherwise, if you go and see the particular sequence or some specific sequence, so there may be multiple CDS and there is a different length. You can find it out. If possible, I will show you after the end of this lecture. I will show you how to see this. So this feature is very important because it contains about the CDS. Here you will get the detailed information about the species or what is it is translating and how it is the it is the translation version of this. So this is DNA and this is the translation version of this. Wherever it's a primer, all of you know for the DNA sequencing you required primers. Okay, so see here it is a primer report. This is the primer bind. So it is the primer information. Okay, and there is a promoter information. We require promoter also. So this is the information, and then the third part is origin. So this is the third part. As soon as you will find origin, there is a beginning of sequence. Here you can see there is a very unique way of writing the sequence. So just read it, count it out: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So there is a 10 alphabets in one block, you can say, and there is a 6 block: 1, 2, 3, 4, 5, 6. The sequence is starting from 1, ending at 60, 61, 120. Okay, so in MCQs, if you now in your syllabus MCQ don't come, but if somebody asks, so it always starts from 161, 121. You will see some other formats where numbering starts means numbering mentioned here like 60, 120, 180, like that. You will see some sequences. Okay, so it is in segment of 6. So it is 2686. So you can see it is 2581 and 90. Okay, and so 81, 91, 2600, and then 1, 2, 3, 4, 5, 6, 7, 8. Okay, so 3 and 3, 6. It is not completed, it is just 6. So as soon as the file get complete, you will not see the termination here itself. In the next line, you will see termination. So this backslash shows the termination of the file. So this backslash shows the termination of the file, and it is not just there where the file ends. No, you will not find here. It is just to the next line, and it is backslash. Okay, so this is about the GenBank file format. I'll just summarize this. It contains 3 main segment: header, and header contains locus. Feature contains the CDS coding segments, and origin, it contains the sequence. So this is very much important. You must remember this. Okay, so this is about the GenBank file format. Next is the EMBL file format. As I told you this also, EMBL is a European Molecular Biology Laboratory. Okay, so how does it file format looks like, and see the difference. Both the files looks almost similar. Only difference is in GenBank it is clearly mentioned definition, accession, version, okay, keyword, source. But in the file format of EMBL, you will find here it is a 2-letter code. Here it is a 2-letter code, and there is a no clear meaning mentioned here itself in the file. So you have to understand what is the meaning of this. Okay, rest everything is same in this file also. Here it's accession number, it is a standard DNA, it's something like Let's see what it is. It is a protozoa. Sorry, it is protozoa 591 base pair. 1, 2, 3, 4, 5. And there is a no date of submission. No date of submission in ID line. Okay. So there is a no date of submission. So ID stand for identification. See, I have given all the meaning over here. You can just pause the video and can relate it. AC is the accession number. Wherever you find the double X line, see, I don't find double X line, any double X line here, but in most of the cases, you will find these double X line after so many. So double X line defines the blank, or you can say space, anything. DT, this DT is a date line, DT. DE is a description line. KW is a keyword. OS is a organism species. OC is organism classification. RN is a reference number. Reference number. RP is reference position. So 12591. It's a complete sequence. It is also talking. RX is reference cross cross reference. Reference cross reference. So from Medline you can also find this one. If you go to Medline and type this number, you will get that same information. RA is a reference author. Who are the authors who worked on this? RT is a reference title. RL is a reference location, and this is RL. RLRARP, same thing it is coming. Then DR, database cross reference. So SPTR EMBL. You can go to this database and you can type these IDs. You can get the information about this particular file. FT is a feature. In that one, in GenBank, it is mentioned feature, but here it is FT. See, it is FT. It also contains about the CDS. Okay, so this is also a complete CDS. 1.1 starts from first sequence to the last one. Okay, so it is a translated table 11, and in the formation of about the protein and all, it's mentioned over here. And SQ is the sequence. Okay, here also, as I told you before, see what is the difference. Here you don't know how many number of A's here, how many number of T's, C's and G's here. But in EMBL you can find 157 is A, 133 C, 170 G, 131 T and 0 other. Sometimes you may find some errors or some extra letter which is not nucleotide, so it is comes under other, and but there is no mention. So this is the advanced feature, you can say. By looking at itself by just sequence, it is 591 base pair. It is mentioning everything, what are the content of this sequence file. Okay, as I told you before, here the beginning starts from 1, but here you can see the file is starting from 60, from 1 to 60. There is no mention of 1, 2, 3. It is just 60, 120, 180, 240. Everything, the rest pattern is same block. Okay, of the block of 10, and there is a 6 columns. Okay, so it is going to sequence. Here also the termination is defined as backslash. Okay, so here itself everything is same, only the pattern is different. So this is EMBL file format. I'll explain one more. It is the main difference. If I'll ask you the differentiate between NCBI and EMBL, in EMBL, so you can say here it is a descriptive file format. It is 2-letter file format. 2-letter file format. Okay, here you can find it is like date of submission in locus, and here is no date of submission. In this one, the sequence content, like what is ATGC number, is not mentioned. Here it is mentioned, you can see here. Okay, other thing is the starting one means sequence start from 1 to 1. Okay, here sequence written as 0 means nothing is mentioned here. It's start in this way. Okay, in both it is termination is defined as a backslashes. So this is the basic difference between EMBL and GenBank file format. Hope you understand this. If you feel any doubt, you just post your query in the comment section. Third one is a PIR file format. This is protein, as I told you, protein information resource. protein information resource. Okay. And then this is also known as NBRF PIR. And what is the full form of NBRF? It was asked once in AKTV exam. It is a National Biomedical Research Foundation. NBRF, National Biomedical Research Foundation, PIR. This is also a very important sequence if you are going into the PIR database and you will download the PIR file to look into the particular amino acid sequence. How does file looks like? Okay, so just see here. A sequence in PIR format consists of one line starting with A dot A. Oh, sorry, it is A greater than sign followed by a two-letter code. Means the file format starts with just like a FASTA format, begin with greater than symbol followed by two-letter code, and these are the two-letter code. If it is P1, it might be greater than F1, if might be greater than D1, or it might be greater than RC. Whatever is the possibility, whatever is the sequence, it is mentioned here. Okay, so greater than PI. Then a semicolon. Semicolon looks like this. Okay, semicolon. Then the sequence identification code, the database ID. Then there is a ID. One line containing a textual description of the sequence. Then it is a description of sequence. What is that particular sequence is? Okay. Then one or more lines containing the sequence itself. The end of the sequence is marked by asterisk. अभी तक जो फाइल्स हमने देखी उसमें एंड हो रहा था बैकस्लैश से, लेकिन यहाँ पे एंड हो रहा है asterisk mark से. Optionally, this can be followed by one or more lines describing the sequence. So, okay. So just have a look of NBR. This PR is also referred to NBR. It is same thing. See, it is greater than N1 semicolon and asterisk 5P210. This is the ID. Greater than P1 5P210 is structure 5P21. So like that, it is start with greater than symbol and there is some particular two-letter code. So what is this two-letter codes are? P1 stands for protein when the complete protein is there. F1 protein when there is a fragment. D1 for DNA, DL for DNA linear, DC for DNA circular, RL for RNA linear, RC for RNA circular, N3 tRNA, N1 other functional RNA, and double X unknown. If there is, I'll remove N1 and I'll write XX. So it means we do not have the information about the sequence and what is that particular sequence is. Okay. So it is just example, like greater than symbol, semicolon, there is a ID and there is a sequence description. What is that sequence? It may be there or it may not be there. That is not a problem. And let's end with, you might be feeling like why these dotted lines are coming. It is a alignment file. It is a alignment file. That's why it is looking like. It is just bring down into a proper alignment. Okay. So there is a different alignment they have performed and they have mentioned just. So this is just an image. If you'll go to that particular format, it looks like same what I have given the order here. The same order you will find in the file format. So this is about the PIR file format. Next one is a GCG file format. GCG is a tool, or you can say it is a package. It's a package just like Emboss. Emboss is a freely available. GCG is a commercial one, and GCG stands for Genetic Computing Group. Genetic Computing Group. Okay. It is maintained by the Wisconsin University. Wisconsin University, USA. Okay. So the comments are terminated with the line starting with 2 slashes. So you just have a look on just GCG file format. You just read it. The first mandatory line that is recognized as part of multiple sequence file multiple sequence format file is the line containing the text MSF. See here, it is mentioned here, it is MSF. The next line is mandatory blank line inserted before the sequence names. So here it is left. Okay. There then follows one line per sequence describing the sequence name, length, checksum. Suppose if you have multiple, see here, it's a 1, 2, 3, 4, 5 sequence are going to multiple alignment, multiple sequence alignment. So see, it's a MSF multiple sequence alignment, 510 type P protein, check 7736. These many checks has been performed. And then there is a name of sequence and there is a termination with the backslashes. Okay, so it is describing, and then follows another blank line. Sequences are interleaved on separate lines with gaps represented by periods. Okay, means see, there is a one gap space. You can see here the this is not properly aligned. See here, it is like this. It is not in a proper alignment. If I draw a line, so both some of the data is going to the right or left. Okay. Each sequence line starts with the sequence name with this, which is separated from the line sequence residue by white space. So this is the space it is given. Okay, name and then. So this is GCC file format. When you will go to GCG, if you are get the chance to work with GCG, I don't think so most of the colleges is having the package of this. So if you get the chance to work with GCG, you have to select the file and it has their own format. It get the you will download the sequence in itself in that particular tool. You will download it. It is automatic converted into that particular format and use it and give it to you written. And if you want to print it, you have to print from there itself. Okay, and then you have to. Otherwise, you will take the screenshots. That is the more only possibility for that. Okay, so now next is the PDB file format. How does PDB file format looks like? It is very important, very typical. Yesterday I have shown you how does PDB data looks like. Okay, how if you go to that particular database, how does it looks like? But today I am showing you a PDB file format. If you open a PDB file, if like 3D structure, what is PDB? Protein data bank. Protein data bank. And whenever you will download a protein structure file, the extension of that file is .pdb. So if you open that particular file in some particular tool or like in a word page or it is a notepad, so you will find, you will see a file looks like this. It is just a descriptive file and you are not able to understand what is here and there. Okay, before this line there is lots of descriptions are there, but that is not of our use. We can understand the sequence from here itself. Okay, so the very first line is a atom line. It is just mentioned atom, 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 and in some here some portion it is mentioned as at atom. It means there is a water molecule. So there is a either it is a atom or there is a heteroatom. Okay. Now the second column is atom serial number 1, 2, 3, 4, 5, 6. I'll come here atom name. Just see the residue name. It is just ALA, alanine. Okay, but see here it is residue name and residue sequence number. Here it is 1, means this is telling about this, but I will come back again, don't get confused. So just look here, it is residue sequence number, means it is 1, means if a sequence contains if a protein sequence contain 200 amino acid, so it means it will go 1, 2, 3, 4 and so on 200. But the atom serial number might be 2000. So don't get confused, there is a difference. This is just talking about this residue name means how many amino acids you have. And just wait, a single amino acid, suppose it is a, this is the basic structure of amino acid. This is the basic structure of amino acid and each amino acid is get changed with this R group. Okay, so serial number means each particular amino acid, how many atoms does it contain. So see here, if it is just 1C, 2C, so like that it is CA means C alpha, CB means C beta. Okay, HT hetero means it is the part of this. HB means a hydrogen bond 1, 2, 3, and C is a carbon, O is oxygen, N is nitrogen. So when the file has been written, it is written in such order. Each amino acid get break down into different atoms. Each how many atoms does a single amino acid contains, it is break down into that particular order. Okay, so you can identify from there itself. So this is serial number. So when you'll open this particular file in real, do not get confused like I have only 200 amino acid, but here it is showing 2500 atoms. So the possibility is there. It may be any number. It depends on which kind of amino acids you have in your file, and that file contains, that particular amino acid contains how many atoms in that particular structure. Okay, so that is the breakdown. That is the atom, atom serial number, atom name, and residue name. Here see residue name is same. After this lecture I'll show you. I'll try to add some or otherwise I'll make some separate video on the live tutorials of these files. Okay, if possible for me, if time will allow. Okay, and there is a uh, see it is mentioned here. That's why I was looking where it is gone. It is chain identifier. Most of the time your protein does not contain only single chain, it contains multiple chain, contains multiple chains, and that is A, B, C. It may be whatever it will, D, E, F, whatever it will be. So how, so the number of chains are defines here, describes here. It is A chain. If there is a B, then after certain completion of A, there will. So it is in an order. First A will get complete, then B will come, then B will get complete, and then C, and so on. The number of chains. So as I described here, what is this residue sequence number? Once the next amino acid, suppose it is serine, SER, here it is SER, so now it will get changed to 2, but this will go to 13, 14, 14, and so this it will go into the same order. Okay. Now third one is a XYZ coordinate. When what is these coordinates are? We have these coordinates X, Y, and Z. Okay, so when we plot a 3D structure of a particular sequence, suppose this is a sequence, and when we draw the 3D structure of this into the spatial, we will see the torsion and all, and we'll place the sequence. So what is the position of that particular amino acid is coming on X, what is the on Y, and what is the position on Z. So this defines the coordinates of that particular amino acid in the 3D space, because that is the 3D structure. So this defines the position. Suppose it is point, it is suppose it is 0, so X means 0 0.2, 0.470, suppose it is 0 0.1, 0 0.2, 0 0.3, 0 0.4, 1, 2, 3, 4, minus 1, minus 2, minus 3, minus 4, 1, 2, 3, 4. So it is 0.2470. It means it is lying somewhere here. Let me change the color so you understand better. So 0.2 minus 2.470. It means it is lying somewhere here. Okay. Next one is a 34.94. So I don't have a space that much. Suppose it is 34. So it is lying over here. Then it is a minus 14, means on Z coordinate. So G is not here. Z is in this segment. So this is positive Z and this is negative Z. So the structure is taking somewhere this particular shape. So this is about the coordinate of this particular amino acid, this particular amino acid position. See, it is almost somewhere on going to the that up and down positions. Okay. Then it is a occupancy. Occupancy means there is a defined space. Suppose this is X, Y, G, X, Y, and Z, and this is the fixed position where this particular amino acid has to come, and it is going to that particular position. It means the occupancy is 1, but if it is going to change the pattern, the occupancy value will get disturbed. B factor is nothing but it is just a temperature. What is the temperature factor? It is, so it is just segment identifier. It is P1, protein 1. When there is a change in that one, it will get changed. Okay, and then symbol. So this symbol is aside from here. See, there is HT1, 2, 3, but here it will only H. CA C H A H C B H H B1 B2 B3 H H H. So whatever it is mentioned here, the first letter of this comes to the element symbol. So this is a very much important file format you have to understand. If you are in bioinformatics and you are working in bioinformatics, some protein modeling or some docking or whatever kind of a study you are going to do in bioinformatics, you must understand this kind of file format. This is very much important. The next file format, and I think this is the last one. This is the multiple sequence file format. Multiple sequence file format. Multiple sequence means when we have n number of sequence. We will see in the second unit about these alignments. We'll read about it. We will solve some numericals also. So when you have a multiple sequence and you have to align them, suppose it is A T G C C A, T A G C A C, and T T G C C A T A, T G G C C A T. So these are the four sequence: 1, 2, 3, 4. More than 2 falls into the multiple sequence alignment. Okay, so let them rearrange them. So if it is A T G C C A, now it is a T A, sorry, it is, there is a multiple way to arrange them. Okay, it is not just simply I can write it is a T A G C one gap C G C A C. Okay, T T G C C T A. Okay, we can make one gap here also. So likewise we can go for the multiple sequence alignment, and over you can see I have to, here is a 3 sequence: human, oyster, corn, and there is a sequence alignment. These dotted lines, they are define the gaps. There is a gaps means they do not find any letter similar at that particular position. Okay, you might be seeing there is a 50, 52, and 60. What does mean sequence is going some less or more? No, this is the number of amino acids coming in that particular range. The main thing you have to understand here is the meaning of these particular colors. See, here is a double dot, here it's a single dot, and then there is a star. What is the meaning of this? So star defines as full conservancy. See here, it is FFF, WWW, WWW, where is a VVV, GGG. So means when the complete data is matching, it is a star. Double star means 2 are matching, 1 is not matching. 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 But don't get confused, always 1 is less. It means they are conserved but not with full confidence, but not with full confidence. And single star means no conservancy, or you can say poor conservancy. Okay, let's see RKE, PPA. Now see here, it is PPA. Why it is not double star? They are coming together, but they are not at all conserved. They are poorly conserved. The tool understand, the methods understands. Okay, so that's the meaning of these lines. So hope you have got the idea about the how the file formats looks like. So I'll just summarize it. I'll just summarize this, what we have studied today. We have got to the file formats and and we have gone through the FASTA. It is just a greater than symbol. Then we gone for GenBank. It is a contain 3 things: header. Header contains what? Locus. Locus contains 6 things that you can go and read. Feature, it contains the CDS and then the origin. it contains the sequence. Okay, then we talked about EMBL. EMBL also contains the same thing, header, feature and origin, but here it is a two letter code. Two letter code, not the complete description is not given. Here we are getting the sequence information, how many number of ATGCs or amino acid sequence are there, we can find it out over here. Okay, just a second, sorry. Okay, then we have gone to the GCG database. A GCG is a tool or package where we have some information about how to use the tool, and then there is a PIR. Okay, there is also a two letter code, there is a meaning of that particular two letter code, and we read the PDB database, PDB file format. Okay, there is so many fields we talked about, and then the last one is the MSA, multiple sequence alignment file format. So this is the detailed information. Hope you have got the idea about the file formats and their requirements and how they will use by the databases, by the tools, or a person how they can understand that file and the output files. So this is the very much important things. If you have any kind of doubt, just put me a comment in the comment section. I'll explain that. If you require the notes for this, just send the comments in the comment section. I'll share the notes with you. Okay, thank you very much. Like you can like my channel, subscribe this channel, and surely share with your friends. Okay, for the more and more your supports I can get from here. Happy learning. Thank you very much.